So welcome back for those who have attended our session before. But I know some of you who probably still don't know me. Always want to introduce myself at the beginning. So my name is Sally Chen. I work at BC Children's Hospital Research Institute and the Women's Health Research Institute with BASPing. We are working under the Digital Health Research Program. Today, I will host this meeting with BASPing, so I will let her introduce herself briefly as well.
Sure. Thanks, Sally, and welcome everyone. My name is Beth. I'm the program manager for digital health research, and I'm excited to be back here with all of you, starting our series for 2025. I'll be moderating the chat, so if you have questions as we go through the speakers, please just put them in the chat, and we'll address them at the end.
Thank you, Beth. So just like said, the seminar will be recorded. So if you miss it or if you cannot watch it all the way through, you can go back to our Learning House corner to rewatch it later on. First of all, I would like to acknowledge that we operate on the ancestral and unceded territory of the Coast Salish people, including the territory of the Musqueam, the Squamish, and the Tsleil-Waututh Nation. We recognize their ongoing contribution to the health and well-being of their community. I also want to express my respect and gratitude for the opportunity to live, work, and serve on this land because of them. It is crucial that we honor and support their right, culture, and history of Indigenous people as we work towards reconciliation and improving healthcare services for all of us. Okay, without further ado, today's topic, finally, is the Big Data Fast Solution, High Performance Computing in Digital Health, aka HPC. It is my pleasure to introduce our three amazing speakers. So Dr. Wyeth Wasserman. Dr. Wasserman is a professor in the Department of Medical Genetics at UBC and based at our BC Children's Hospital as well. His computational biology research focuses on the analysis of the human genome. His research laboratory at BC Children conducts research in two overlapping domains, the control of the gene activity and the identification of the causes of rare genetic disorders. His lab created JASPA database and a wide range of software which are used globally to identify the on and off switches in gene. Karlie Edwards. Karlie Edwards is a Ph.D. student in Michael Copper's lab, where she is studying the role of epigenetic in immune system development. Since receiving her bachelor in biochemistry, she has worked in fermentation sciences, infectious diseases, and is now utilizing bioinformatics to understand biological underpinnings of health in children. Last but not least, our first speaker today, Jonath Sujan, our Iris Business Operation Manager, will first give us an overview of what is high-performance computing. So, Jonath, the floor is all yours. Why don't you start by introduce yourself? I will stop share now.
Yeah, thanks, Ellie. Hi, everyone. It's a pleasure to be here. I'm Jonath Sujan. I'm a Business Operations Manager for the Integrated Research Informatics, or the IRIS team. So for those who don't know about the IRIS team, the team offers a wide variety of services, which ranges from data management to managing a variety of IT infrastructure that is used by the entire research institute. And we also have the digital health research and the innovations office under it, which does a lot of variety of things around data analytics, AI, model developments, and a lot more. So today's topic is around big data and what kind of solutions and facilities we have available to support these kind of initiatives. So we'll be talking a little bit about what big data is and how to use high performance compute facilities, what facilities we have here, and how all these things come together to enhance a variety of research activities and innovations happening within our research institute. So what is big data? So I don't think we have a very set defined metric saying that it has to be an X amount of data to be considered as a big data. It's usually a data set that is like fast accumulating. So large amount of traffic, large amount of data that is being accumulated over time. It could have like complexity to it. For example, the data we collect here in the hospital, it can be structured, it can be unstructured. We have like genomic data collected, we have like medical imaging data collected, a large variety of data. And it's usually a large data set. So we usually consider like those in the range of like gigabytes and above to be considered a big data. So we see big data kind of like everywhere we look around. So if you have an electric vehicle, it has a lot of sensors in it. It collects a lot of data to make it function. So when we go into the Internet of Things like realm, we have like home security, a lot of sensors like communicating all together to make things possible. And also a lot of traffic going through the internet whenever you use the internet. So those are all like tracked to some extent by the internet service provider. That becomes an accumulation of data. But what are some examples of like big data in the digital health or the healthcare realm? So there was a recent like report released by the RBC, which says like 30% of the entire like world's data, it's actually generated by healthcare systems. So this could again include a variety of things like genome data collected for several purposes, whether it be research or clinical activities. So when we do the sequencing, genome data can easily generate like gigabytes. And the advanced like sequencers, they can sequence like multiple samples, so which could easily result in terabytes in a single run. The medical images that are being captured by our advanced scanners, again, like they can generate like gigabytes of data, like high quality data. The electronic health record system that is used by the hospital and clinics within our hospital. For a hospital of our size, like it can easily like generate like gigabytes or maybe even like terabytes. We also have like several physiological like recordings happening with the patients, like EEG, ECG, a lot of like patient monitor like related data that are being captured. Those can also contribute to a big data. Finally, if you have any like like wearable devices, they also have like a lot of sensors in it. They capture a lot of data. So if we stream them into a particular system to be used, it can also be considered a big data. So there are a few characteristics to be considered as a big data. For example, we call them as like 5Vs. First one is the volume of data generated. So like we mentioned before, it's usually in terms of like gigabytes. So it should be a huge amount of data. And the velocity at which the data is being generated or being captured. Again, like keeping in mind the electronic health record system for one single patient, it could be a small amount of data. But when we think about the entire like an organization or a hospital, like collecting data from multiple like patients and putting them into a system, there's going to be a lot of data that will be collected. The variety of data that is being generated, even when we think about the EHR, there can be like structured like data fields where data is being put into. We also have like medical notes or doctor notes, which has like unstructured data in it. And the two important thing I would say is the veracity or the quality of data and the value of data. In the next few slides, we will talk about a few considerations for if you're planning to set up or start a big data initiative. It's going to be a huge investment at the very beginning, so it is important to keep in mind the quality of data that's being generated and what the actual end goal is or what value that data brings. So if you have plans to collect a large volume of data, or if you have like initiatives to like merge a lot of data together that results in a large volume of data, like how do you do that? What should you keep in mind? First and foremost, like define a proper data collection protocol in a short period of time. Like if it is properly set and if it's collecting a variety of data, it can like grow really fast. So changing like certain like data variables within the data set in the middle of data collection can be really tedious, and it will also affect the data quality. So like decide on something that will be useful for what you want to use it for at the very beginning. Also have an efficient data management like plan. So if you're using any like databases, make sure it's like columnar, if it is like optimized for easier retrieval of data. And sometimes for analysis, you might want to take like a large volume of data out. So try to index them and cache like data that you frequently access. An important thing is like secure storage. Make sure the storage you use has like backups, it's durable, it is scalable as we go, and make sure the systems you use for the collection is like reliable and the network throughput. There's going to be a lot of like data movement happening, so make sure the network is able to handle it. And also, if you plan to integrate them all together, make sure the data is interoperable. And finally, once you collect all this data, you need to perform analysis on it. So make sure you have access to high performance computing services to do that. So what BCCHR offers, so we have like secure storage, we have like high availability servers. Our network can support like several gigabytes of network traffic. And if you need to transfer like data between systems, we can also like help you with that. If you need help with like an analysis or AI model development, we can help you with that too. And we also have like our in-house like general purpose compute cluster, or HPC, to help you with processing and analyzing these large data sets. So moving on to the next section of the seminar, like what is like high performance computing? So it could basically be a powerful computer or a cluster of like an interconnected powerful computers that work together to perform a series of operations, to perform a series of like complex calculations and analysis at a very high speed. So whenever you want to analyze something, some large data set, you might not be able to do that in your traditional workstation. In that case, you need to like use any HPC facilities you have. Just in terms of like how powerful these computers could be, say any kind of computations that can take like years to do in your like workstation, these high performance compute servers can easily do that in hours or days. So briefly talking about like general purpose compute cluster, which is a high performance compute facility we have within BCCHR. So it has like several like compute nodes. So some of them are dedicated to specific research teams, but a lot of them are also available to the default like queue, or accessible to general users. Like anyone can log in and use it. You just need to approach us to get access to it. The way it works is like you just submit a job of some kind, and whenever a compute node is available, the job gets like executed in a compute node, and the results become available. So all of the compute nodes we have are like 384 gig RAM with like 40 cores of CPU, just to like again give a comparison against your workstation. If you have like a 32, most of the workstation you use would be like a 32 gig RAM with maybe like 4 to 8 cores, but these are like very powerful than that. So some tools that are like basically like already available within the GPCC. So in order to facilitate like executing these jobs, we have like Slurm. Most of the analysis it happens using like R and Python. So we also have some IDEs to interactively develop these scripts. We have like Conda in case there are certain tools you want to use that is not like available natively within the HPC. You can install them within a virtual environment you create. We also have like an integrations with the Compute Canada like software like modules. Any modules you might have, if you had already used like Compute Canada, and if there are certain modules you're interested in using, you can use that from within the GPCC. And we also have our own like software package, and there are a few other like basic tools. Also want to quickly walk through some of the enhancements we are planning to the GPCC, like because we are seeing a lot of like big data sets collected, and a lot of need for these HPC facilities. We have been working closely with the systems team, with the BCCHR, and also the IT team to increase the number of like compute nodes. We have like made some relaxations to allow like parallel computing using multiple nodes and to run like several jobs. We have purchased like small like low-level like GPUs to see if there is demand for those kind of activities, because a lot of the AI initiatives are starting within our campus. So we want to support them too. In the long run, if there is more demand, we'll purchase like more powerful GPUs. There are plans to upgrade storage to have like long-term storage with the HPC, and some OS upgrades and GUIs to to enhance the security of the service we offer and to add more functionalities. So I think that is all I have. So if you would like to know more about the GPCC or big data initiatives happening within our campus, feel free to contact the DHR office or just send us an email to iris@bchr.ca. Happy to have more conversations. Yeah.
Thank you so much, Jonas. That's an amazing introduction of the big data and the HPC. If you're okay, could you leave the IRIS contact info email in the chat? So if people have questions, they can reach out to you directly and your team.
Yeah, absolutely.
And Dr. Wasserman, you can share your screen. Thank you. Oh, you mute yourself.
There we go. I think you can hear me now.
Yeah.
Hopefully you can see slides.
Yes.
Thank you, Jonath, for the nice introduction, because we've done all of our computing on our project on the GPCC, and I think we've figured out every way to break it along the way, but it's been amazing the support that we've had to work with very sensitive data. So I'm going to take a few minutes to tell you about the Silent Genomes Project, which is a project that I've had the pleasure of working with Laura Arbour and Nadine Caron on for the last now 7 years, as we've been confronting a challenge of equity for Indigenous peoples in terms of rare disease diagnosis. So I'll take you a little bit into this project, and it's an unusual project in the sense that it's the largest compute project that I've worked on in my career, with massive amounts of data and servers, and that's the easy part, because it's confronted with enormous challenges in terms of the governance and social side of the project. So I'll just give you a little bit of exposure to that as we go through. I will reiterate Sally's introduction and say that we are deeply grateful for all the support we have. Our elder on the project is from the Squamish Nation, and the support has been incredible from the communities. So when you're doing rare disease diagnosis, and many of you on the line here, I notice, are very familiar with this type of work, you'll have some patient who arrives to a clinical setting. A clinician will do their very best to gauge and understand what's going on with that patient with the rare disease. If appropriate, they'll go and order a DNA sequence to be generated on that patient, and then that DNA sequence gives you back a list of variations that are observed in that patient. And the traditional model is to then compare that list of variations that you had against a database of variations that have been observed before and say, okay, these ones have been seen in a lot of people, so they're probably not causing a rare disease. And that eliminates most of the variations that you find, and then you can focus your attention on the rest. And so when you do that, it's not uncommon in your sequencing an individual to come up with about 4 million variations from the reference genome when you're doing this. And you apply your filters on there, and you might take it down to a couple 100,000 in the set with that sort of variation piece, and then you can use some additional approaches to reduce it further by considering the family structure, what's present in the parents, other siblings, anyone else you have in the system, various analysis tools. And when you get it down to that small number of variants, then the experts get in there and really start digging around and trying to figure out which of these might be going on. That can take a lot of time, and it's challenging. That's in the sort of standard case. But back in 2016, in the clinical rounds, I was listening to the description of a case where this didn't work right. And what turns out is that when you look at the reference data that's in Nomad, it's not equitably distributed. This is the current version of Nomad. And what you're going to notice is that you have some populations, like Europeans, robustly represented in the database. 77% of the 807,000 sequences there are from European-derived backgrounds. And you'll notice that the remaining individuals is sitting there at a little under 4%. And remaining individuals is all sorts of populations of people around the world. And when you look at Indigenous peoples of Canada, it's almost zero. So when you go and try to do this type of analysis with the removing that. You do get some benefit of the databases because there's a lot of shared variation across all humanity, but much of the variation that you find in when you try to go there and do the work with the indigenous peoples is installed. And so instead of getting sort of a big reduction down to 200,000, you might get a reduction down to 600,000 with the available data that's there. And then you might end up with 30 or so. And as I said, this final step can be very challenging. And so what you end up with is you can't distinguish what's potentially causing the disease from what's not. And you end up with many patients stalled in the diagnostic process and not getting a speedy diagnosis in what they're doing. Now, the work of this kind is really challenged because we have a deep history of bad actions in working with indigenous peoples in terms of genetics, and that makes it so that there's not trust and not a history of working well with four communities. And that's led to a lot of global considerations about how we do better with handling indigenous data. And just to be clear, these types of actions continue to this day. So it's not like we're talking about a historic problem. We continue to see stories popping up even today where we have these types of events happening. Now, certain practices have been, and policies and approaches have emerged in trying to confront how we can handle data safely when working with in partnership with indigenous peoples. And for First Nations peoples, the OCAP principles have been robustly developed. And that sort of speaks to the idea that ultimately it's nothing about us without us. So ultimately data belongs to indigenous peoples. It does not belong to researchers. The data needs to be under indigenous oversight, and it needs to be handled in appropriate manners with well thought out governance and processes. Now, in Canada, we have three populations of Indigenous peoples, not just First Nations. We also have the Métis and the Inuit, and all three communities have worked with us on silent genomes. And so we also pay close attention to the CARE principles, which are essentially similar to the OCAP principles, but broadening out there, but with many of the same concepts of how we can safely handle data in terms of Indigenous data. So we launched into the Silent Genomes Project to say, could we create a reference database? And ultimately, that conversation centers on governance first and implementation second. And so a lot of the first 5 to 6 years of work were very focused on activity 1 here, which is around the governance formation. And really most of the data processing and development, once we had that governance in place and approvals, took place in the last 2 years. This is sort of in the earlier stages of the work. We had an Indigenous steering committee called ESCRD come into form. We've recently transitioned in the last month to a governance committee, which is slightly differently composed. I just haven't updated a slide for the new governance committee, but the concept of it is it's an Indigenous committee. It's made up of partners who are coming together, and very powerfully in this process have been communities members with connections to genetic conditions there, and that's really means families and people impacted by genetic disorders, and they've been really powerful voices in this conversation to make sure that we consider how to do things safely while making sure that kids who need a diagnosis can get a diagnosis. Everything revolves around that governance. We basically had steps where we could not proceed until the governance was endorsed. And what we ended up doing, and I'm not going to go through all the details of this in the short time we have here, is we partnered with, in the initial stage, 4 First Nations across Canada who agreed for their samples to be used in doing it, providing us geographic distribution. That led to a set of 600 DNA samples being provided for this work. And this was done with approval of the health committees and the nations for the samples to be used for this purpose. The governance led to a decision that the data should be available for diagnosis. So to be enabling diagnosis, it's not a research tool, it's a diagnostic tool, and that it should be under controlled access. So something that would have people who have a need to access diagnostic data should be able to register and obtain access to it, and said that this access should be for anybody who's doing diagnosis who needs it for Indigenous peoples, and not restricted to those in Canada. So it's available to people in the United States and others. What's in this database? We do not keep any identifying information about the individuals. There's no health information. We don't know anything about the health properties of these individuals. All we have is the raw genetic information. So we basically can say at a given position how many people have an A at this certain position, how many people have a C at this certain position. And so it's just that raw population scale data that says how many have it. So speeding through this a little bit, the 600 samples were sent to the Genome Sciences Center, BC Genome Sciences Center. They did paired end Illumina sequencing for this. This is just an example of the fold coverage. To give you a sense of scale for high performance computing here. Each of these individuals was done at 30x. 1x is 3 billion base pairs, so 30x is roughly 90 billion base pairs of sequence data, 600 individuals, and you can see where Jonath would have been stressed out by us coming down there, because it's very easy for us to sort of just start taking up terabytes and terabytes of storage. And when we go through our analysis that I'm going to mention in the next couple of slides, it balloons out because we have to sort of do a lot of processing. And so at the peaks, I think we were up to about 120 terabytes of storage that we were occupying to handle this data in processing. We implemented our analysis pipeline using a system called Nextflow, which is a pipelining software system that's emerged as widely supported around the world in the last few years. And we've had to implement a variety of tool components within that. So a lot of work around the world has been developing specific tools for specific aspects of taking these puzzle pieces from these genome sequences, mapping them onto the reference genome, then doing analysis of those mapped on reads and trying to say, is there a letter change here or not? Is there a structural variant change here or not? And so on. And so it takes us about 2 months to process, about 3 months to process all 600 genomes through our pipeline. So just to give you a sense of large dedicated nodes on the GPCC churning nonstop for about 3 months to generate the reference library. And currently, what we found is that we have 26 million total variants observed across the individuals participating. That compares to Nomad, which has close to 1 billion variants observed. But only 80% of the variants we observed in the First Nations samples that we worked with here were previously observed in Nomad at all. So 20% of the variants that we're finding are completely novel, have not been observed before, and that's striking. This is just to give you a sense of how frequencies vary between the Nomad and the IBVL. And what you're going to see is on the upper left, and the density here is hard to detect, but on the upper left, you see those things that are in the IBVL. And this is just looking at chromosome 22, the smallest chromosome. So it's just giving you a small taste of the whole. So last month in January, we were thrilled that we could actually release the IBVL for use by clinicians doing diagnosis. And so we've been gradually enrolling and improving users onto the system. We now have about we're slow rolling it out. So we have about 30 users spread across Canada right now, working with the database and the system and using it for their diagnosis and their clinical teams. And that's been greatly enabled by work we've done to have a national network of clinical genetics connected together working on this. And I'll speed past these slides just to say that you can picture just how large a project this is when you're trying to work through engagement with community, sequencing large numbers of genomes, building out a national network of clinicians and engagement. And so I'm grateful for the enormous amount of support we've found all along the way. And so it's been absolutely incredible seeing people willing to find ways to make this work in a project that is very complex to come through the internal team and the funders. So thank you very much for your attention, and hopefully I saved some time for Karlie.
Thank you. Thank you so much, Dr. Wasserman. That's fantastic talk about the your silent genome project and IBVL. I'm happy that you got the result published in January. And definitely a lot of teams collaborate together, not a small project. A lot of collaboration are needed. We encourage everyone to share your comments and questions in the chat, and we will during the last 5 to 10 minutes Q&A to go through your questions, or later on you can unmute yourself. But without further ado, I will let Karlie to talk about her project. Thank you, Karlie.
No, thank you. And thanks, Dr. Wasserman. It's always great to hear what you're doing with the Silent Genomes Project. So I'm going to take a little turn, and we're actually talking about DNA methylation and the work that's going on in the COBRA lab. So how we're leveraging high dimensional data to uncover what's happening in terms of epigenetics, and how I'm looking at that in the terms of healthy pediatric development. So just kind of give you a little overview. So the COBRA lab studies epigenetics, which are molecular changes that occur around the genome. So they don't affect the underlying sequence, but they do change the way genes are expressed. So this can alter cell function and cell type identity. We do this in yeast to look at histone modifications, and we study this in mammals to look specifically at DNA methylation. And DNA methylation is probably the simplest of these. It is the addition of a methyl group to the cytosine residue within our DNA. And this typically occurs when a cytosine is next to a guanine. So these are called CPGs. And when we have increased DNA methylation in the promoter region of the gene, we typically associate this with decreased gene expression. So you can think of those methyl groups sticking off the DNA as like a biological stop sign that blocks transcriptional enzymes from accessing and reading the DNA. Conversely, if we don't have DNA methylation in the promoter region of the gene, we have access to the DNA to read and transcribe the genes, so we associate that with increased gene expression. So we do see changes with DNA methylation and function, and then gene cell type identity, depending on where the mark is. I'm really interested in studying this in pediatrics, but we are studying this kind of across the life course in the COBRA lab. So we have ongoing work using birth cohorts to really kind of capture early childhood development, looking at midlife to see kind of what maybe reflections of experience that happen in early life you can measure in that mid to adulthood, and then looking at aging populations. DNA methylation does change predictively over the life course. So this is something that we can kind of see and look at as we age. So how do we get our data? So this is where the HPC part really comes into play. So we take our DNA, and then it undergoes what's called bisulfite conversion. So that's like a chemical little protector of that methyl group to make sure it doesn't get lost in terms when we have the DNA replicated before we move forward with it. Once the DNA is bisulfite converted, we use the Illumina chip technology for our analysis in the COBRA lab. And so I'll be talking about the EPIC V1 chip right now, but there were previous iterations, and we have since come out, they're working on EPIC V2 right now. The EPIC V1 characterizes 850,000 of those CPGs within our human genome. And that's actually only about 3%, but it's a really good coverage of all of the chromosomes, as well as genes that are found to be important in different mechanisms and diseases throughout the years. So we have a really good picture of what's happening over the course of our epigenome-wide changes. Once we have all the DNA on those chips, they look like this, and they are amazing. Team of staff technicians in our lab go through different stages to kind of assemble these chips with spacers. They get stained with different chemical fluorophores, and then they go into this little eye scan instrument that then excites the fluorophores. So we have green is associated with methylation, and red is associated with an unmethylated CPG. So we have all of these little light intensity files that come off for every single sample for every single data point. So you can see how this is not at the same scale as Dr. Wasserman's data, but this is still about 850,000 data points for every single sample that we run. So we have definitely needed the GPCC and the HPC in order to process our data. I am really interested in studying this in the context of the immune system. So that's what I'll be talking about for kind of the work that I've been up to. And so just to kind of frame the immune system, we have the innate immune system, which is our immunological starting place, the first line of defense when it comes to foreign invasions and bacteria and viruses. And we have the adaptive immune system, what changes as we get exposed to things. All of these cells, to some extent, have the ability to change identity or differentiate, and they have the ability to perform different functions when the need arises. Both of these are hallmarks of DNA methylation and the ability to identify cell type and change functionality. So that's why I think putting these two pieces together is really interesting, specifically in the context of early childhood, because we have this rapid change happening in the immune system as kids age from birth to 5, and understanding the role DNA methylation plays in epigenetics and changes in cell type identity and function, I think is really interesting. So I'm studying this in the context of the developing pediatric immune system. And I have had the amazing privilege to work with the CHILD study. So this is the Canadian Healthy Infant Longitudinal Development Study, which started around 2010 with 4 enrollment centers nationwide. So we have a really good representation of the country. Women were enrolled during pregnancy, and we have ultimately about 3,200 mother-daughter or mother-child pairs that are enrolled that we have been collecting data on. This resulted in 47% female infants and about 72% with European ancestry. So we are still a very predominantly European ancestry cohort, but we are very fortunate to have some diverse representation of indigenous populations and Asian, Indian, et cetera, which is very nice compared to a lot of the European cohorts that are similar in terms of like collection data points that are predominantly 98 to 99% European ancestry. So this is a really powerful part of the study, and it's been very well phenotyped. So the data I'm talking about is just a small fragment of what's available to really ask questions and understand what's happening in these first years of life. The samples that I'm working with in particular are the DNA methylation ones. So we have about 760 kids that had whole blood collected at birth, age 1, and age 5. Those samples were, we collected isolated the DNA, ran the arrays, have our DNA methylation data. So we ended up running about 2,400 samples. And then from there, I can look at cell type proportion, as well as some really cool things like epigenetic age. So to get kind of started, I was interested in immune system, cell type proportion. So we were using whole blood, which is a heterogeneous tissue. And although there are methods that we can look for granular cell types like flow cytometry or like complete blood cell counts, that requires additional time and resources. And so with DNA methylation, we can actually take the signature that we get from the whole blood and use reference panels to bioinformatically deconvolute the cell type proportions. And that gives us kind of this picture of what is happening in terms of changes in cell type proportion in early life, just from that DNA methylation data. And what was really cool and reassuring to us is that we saw this increase in neutrophils from ages 1 to 5, which has been captured in the literature multiple times, as well as this normal decrease in naive cell type proportions as we're gaining memory with exposures and vaccines, et cetera. So we were really excited to be able to capture this incredible remodeling of the immune system using our DNA methylation data. And given that we see these significant changes in all of the 12 cell types that we could find, I was really curious to see kind of what that meant in terms of DNA methylation. So in order to do that, I do an epigenome-wide association study. I didn't do epigenome-wide in this case. I focused in on immune genes. And so I was looking for associations between DNA methylation in immune genes and age. What is happening in terms of our epigenome between ages 1 and 5? And surprisingly to me and my lab, there were actually very few changes. Given how much remodeling is happening in the immune system, I think we expected to see quite a few more changes in DNA methylation, but we saw 10 sites that changed between 1 and 5. But what's really cool about these sites is we have these handful here that are associated with the bacterial response, and these other handful of genes here that are associated with viral response and interferon pathways. So seeing these change between ages 1 and 5 really makes sense in the context of, again, kids going to daycare, school, getting exposed, getting sick. So seeing these changes happen in the first 5 years of life really made sense in the context of early childhood development. One of the other concepts that's really interesting and kind of really big in the field of epigenetics is this idea of epigenetic age. And so we have our chronological age, that's our calendar, take the year you were born, that's your age. But we also have our biological age. And this can be measured in a variety of ways, including through DNA methylation. So we can compare our epigenetic age to our chronological age and kind of get an idea of our overall health. And so if you are in line, you're concordant, that's great. If your epigenetic age is younger than your chronological age, that's That's generally pretty good. If your epigenetic age is older than your chronological age, that's normally associated with poorer health outcomes and all-cause mortality. And that's really seen in adult and aging populations where epigenetically older individuals normally experience trauma, stress in early life that's reflected by that older epigenetic age. And then it's further associated with increases in diseases, normally earlier mortality, and those kinds of things. But this is normally applied to kids as well. If they have an older epigenetic age, they're probably not as healthy, but we don't actually know that part. And so this is what it looks like in child, because I want to know actually what does epigenetic age mean in kids. So we have our calendar age on the x-axis and our epigenetic age on the y-axis using one of the very common epigenetic clocks. And we see that there are definitely some kids that are epigenetically older on both sides of ages 1 and 5, and some that are epigenetically younger. So my question here is, if we have these time points and these epigenetic ages, what does epigenetic age at 1 mean for health at 5? So if we have these kids that are epigenetically older, are they actually more sick, or is this reflective of maybe a maturity that we're seeing it's actually protective? And then conversely, we have the epigenetic age at 5. What does that reflect that might have happened at age 1? In adults, we normally associate it with stress and trauma, but were the kids maybe sicker at age 1, which has increased their epigenetic age at age 5? So these are the questions that I'm working on answering now, because I think this is a really interesting idea and wanting to see how concordant our conclusions are between adults and these aging kids. So that's really looking at, is there a difference between like chronic illness and acute illness? Is DNA methylation age being older, protective, or is that indicative of a specific health outcome? So that's some work that's ongoing. So I don't have any new data to share there. But ultimately, I think DNA methylation is an incredibly powerful tool that really allows us to dig in and understand biological embedding of experience, and it allows us to potentially establish trajectories for future health. We are able to use this data to predict cell type proportion, epigenetic age, and even more. There's predictors for smoking, there's predictors for cytokine concentration. So there's a lot of different tools out there that use DNA methylation data to kind of get a full picture of the underlying biology. DNA methylation in my context was actually relatively stable in immune genes in early life. And I've done some more work in exploring the immune system and what's happening in these first 5 years beyond what I've showed today. But I think it was interesting that DNA methylation is not the main regulator of development in terms of early life immune system. And there's tons of data here, which is why I am really grateful for BCCHR and the GPCC to be able to actually do these analyses. Like Wyeth said, these take a long time. And if we had to run them on our local machines, it would be nearly impossible, and it would take so long. So this has really made our work possible. And I think that child in particular allows researchers to ask a lot of questions. So we're really fortunate at BCCHR. Dr. Stuart Turvey is the Vancouver site lead for child. So we have this amazing resource like in-house. And if you're interested in child, they have collected data from environmental, nutrition, different biological samples as well beyond the whole blood that we've used here. So more information can be found at the Child Study website if you're interested. And with that, I'd like to thank you again for having me here today, members of the COBOR Lab for being incredible mentors throughout this journey, the Turvey Lab for their support with all of the child stuff, and the rest of the Child Study team and family participants. So I'm happy to answer questions during the Q&A about any of this work, child, or anything else. Thank you.
Thank you so much, Karlie and Wyeth and Jonath. That as you're speaking, I was really reflecting on the new strategic plan at BCCHR and how digital health and data analytics is an strategic enabler of research. And I think this is some of the greatest examples I've heard recently of how these tools and our capacity to use data as it grows is really enabling this amazing research and generating insights that have real impact on people's health in BC. So thank you for your work. I wanted to just remind people, if you have questions, you're welcome to put them in the chat. If you want to raise your hand, also happy to have questions there. But to get us started, I'm also happy to ask questions. So, Wyeth, congratulations to you and all the team on that work. I haven't heard the update on the Silent Genomes Project, but what an incredible journey you've been on in the last 7 years to get to where you are. I did I did notice in your original slides about just the coverage of that main genomics data set that there are many populations that are missing. And I wonder if you could reflect a bit on the lessons learned if you were to take the work of silent genomes and think about expanding coverage to one of the other communities that is underrepresented. What would you say to trainees or researchers that would want to embark on that process? What are the main lessons learned you would share? Because it seems like there's opportunity to continue this work, and I'd like to encourage that.
Yeah, so there's a few layers to that. I'll try to answer with a couple components. So one is I had the joy of spending a sabbatical year with the Maori Kakano Variome project, which is very similar to the Silent Genomes project, and we've worked very closely. And so we do see this type of good work happening multiple places around the world, and we learn a lot from each other. And that's led to the formation of the International Federation for Indigenous Genomics, which is meeting later this year, and so to continue the work to promote that. So there is global commitment to this space, which is very heartening to see, and it's amazing people in many places that we're getting to know. When you think about Canada and you think about where we are there, I mentioned that we had 4 First Nations that participated in the initial release. There are more than 600 First Nations in Canada. There are Inuit populations and Métis, and the diversity is incredibly huge. And so we do not think that we are done. We think we have a beginning in terms of what's there. And one of the things that we're exploring is how can we empower communities to gauge how good the IBVL is for their needs. And so we're doing a fair bit of research right now on trying to think about how many samples would a community need to sequence in order to just get a gauge as to whether the IBVL is going to serve their community well, or whether they would benefit potentially from participating or creating their own resource, depending on their perspective. And that's very exciting because it's leading to an opportunity to engage more broadly. And we're looking forward to sharing that with some of our partners across the country over the next little while. Also, in the Canadian context, there is something called the Pan-Canadian Genome Library, which Silent Genomes is participating in. And it's complex to make sure that we maintain Indigenous data sovereignty over data in that setting. But that's led to a number of other projects that are potentially going to be exploring opportunities for data to be contributed towards silent genomes or other Indigenous governance mechanisms. So lots of good work. And I don't know if I answered your question there, but I just sort of conveying that there's tons of stuff happening.
No,
and
it
amazing
is
work.
great. It's great to hear how connected this is to the global efforts, and more efforts to expand the communities being included across Canada. There is a question in the chat. Before I get to that, I just wanted to ask Karlie one question too. Thinking about this idea of digital health and data analytics as an enabler, you have so many different types of data that are relevant to your project, and ways of collecting that data are also probably diverse. Can you just speak to some of the resources that were required to support the child health project, and how you see digital health tools factoring into that?
So yeah, there is a wealth of data, and we are lucky to have Stuart and team in-house. So the DNA methylation data and the genotyping data were processed all by our lab. So that was something that I was able to be really hands-on with and kind of see throughout the process, which definitely would be different than some of the labs that might be requesting that data raw or finalized. And then we had the microbiome data, which is available as well, and that was produced by the Turvey lab. So we at least have like those data sets that are like in-house, easily accessible, and with the people that are very hands-on with the data. But there's other data sets, like there's Olink, which gives us the idea of like cytokines. And so being able to reach out to the labs that process that data so they can help inform on like what the data format looks like, kind of what the parameters were. It's a very, very collaborative, it makes it a very collaborative environment, which has been really great with this cohort in order to be able to work on integrating all of these really in particular data sets. The questionnaires, luckily, are consistent across all of the recruitment centers. So we have consistency in the data formatting that's coming in for like the number of infections, vaccinations, all of that is at least like very consistently formatted. So it all goes through the National Coordinating Center for a Child and comes to us, and we are able to use that data.
That's cool. The two very wealthy big data sets. Anna Meredith did have a question in the chat for you as well, Wyeth, asking, you mentioned that the IBVL does not have linked health data. Can you speak to what the reasons for that are?
Yeah, so just putting a link in the chat for an extra piece here. So it's very complex to make progress in figuring out governance. And there was a sort of perception that we could make progress on getting the general variation data for diagnostic and then move on to other things. But one thing that's very important for this particular data set is that all 4 First Nations that contributed did so in part through their participation in the Canadian Alliance for Healthy Hearts and Minds, which is a population cohort study that's been going on based out of McMaster and PHRI. And there's an Indigenous arm within that that's been going for about 14 years in their work. And so all the data that was generated for the IBVL was also returned to the nations that provided it and is available with linked data through the Alliance for Healthy Hearts and Minds project. So in this case, the data samples do have the potential for that linked health data work to be there for those communities to approve and control in their mechanisms. But the long term is, I think, well aware, particularly with the Pan-Canadian Genome Library, that we will eventually have to get to the point where we have resources that are health data linked, but it's going to be another probably 7 years of conversations to get through the governance of that.
That's amazing to hear. Jonath, to kind of conclude us, I wanted to just ask you, you did allude to the various services at BC Children's and Women's, and we do have an event next week if people are interested in learning more. Can you speak to Iris Day and the other teams, maybe?
Yeah, absolutely. Yeah, so we have our annual Iris Day happening on the March 4th. So where we just present the different teams and the different kind of projects we have supported and the services we offer. Like I mentioned at the very beginning, like Iris is a bigger team. It has a lot of sub teams under it. Data management is one of them. So IT is another, which offers like a wide variety of services along like development, system administration, and like supporting all of these like an IT infrastructure for the entire research institute. BCCHR and the innovations team is also under it, offering services on data analytics, visualization, AI model development, and we're also collaborating well with PHSA and other research teams within the BCCHR and WHRI to support a variety of these activities happening. So if you would like to know more about what Iris is, who we are, and we offer, there should have been a communication sent out. But if you have not received it, contact us. Happy to share the invitation with you.
There is one more question in the chat. Does Iris have access to PACS data? Do you know the answer to that, Jonath?
Unfortunately, we do not have like direct access to PACS. As of now, like so far we have not like received any request or projects requiring like direct access to PACS, but we have collaborated with like other CURS services like the 3D MRI facility to access some of the MRI images and do some analysis on it. But if there are any requirements around like wanting access to like PACS data, like let us know. We'll see what we can do.
Yeah, I'm always happy to try to figure out things that have not been done before, because there's a lot of development in this space happening, a lot of new needs. Any other questions from or last comments from our speakers you wanted to share? I'll give it back to Sally.
Yeah, while we are waiting, like probably just a couple more minutes, if anyone has any question, please feel free to jump in. But I just want to mention for those who attended our seminar, we always have seminar every other month. So the next one is in April. The date is to be determined, but it will be about change management in healthcare. And so stay tuned and then scan here if you want to watch our past learning house digital house seminars about like the AI, the machine learning. We have conducted a lot of those topics in the past. And also you can follow us on Eventbrite to not miss our next digital house seminars.
Thank you once again to our speakers. That was a fantastic update about some of the incredible work in genomics on the campus. And thanks everyone for joining us for the start of the 2025 seminar series, and look forward to seeing you in April.
Thank you, everyone. We'll end